Dear students, good to have with me today. My lecture for this week in human histology, muscle tissue, and nervous tissue. So, all right. Um, there are three varieties of muscles. There are three varieties of human muscles: as skeletal muscles, smooth muscle, and cardiac muscle. Skeletal is like a saturated muscle. Is the most abundant type of the muscle. Is more than 400 distinct muscles distributed around all the human body, and also it's only muscle under a volunteer control, and it's represent approximately 40% of the body weight. While the second one is smooth muscle, it's like manage the contraction of non-volunteer muscle. Well, actually, it's found in every organ system started from creating vascular resistance to uterine contractions. While the cardiac muscles, it's not volunteer control, and because it's along with the heart wall, so it's have a character like it's unique in work and it is automaticity. And it must be unique in work. Imagine if you have one part of your heart is work and the other part is stop. It will be horrible. So it's the essential character for cardiac muscle must be unique and automaticity as a work. All the three muscles have to convert the chemical energy into mechanical work and movement. Right. Now we have another slide about functions of muscle tissue. It's I put all the functions in one slide: producing body movements, stabilizing body positions, regulating organ volumes. It's part of a smooth muscle, and we know that it's regulating the volume because smooth muscle is not found as a single. It's like bands, and the bands it's called sphincters, while the movement of substance within the body, and these substance either liquid or solid or gas, blood, lymph, urine, air, food, fluids, and sperm. While for producing heat, it's involuntary contractions of skeletal muscle. Let me explain what does that mean. If I have this muscle producing heat, we know that each muscle in a human body is like motor, and then because the muscle cells receive the signals or impulses or stimuli and start to relax and contract in involuntary manner, so lead to generate heat. All right. Now, the properties of muscle tissue. It's this slide is essential to know that these properties it must goes like this sequences one by follow. You cannot change the place because if you change this, it doesn't make sense. I will start by excitability is respond to chemicals released from nerve cells, and conductivity is ability to propagate the electrical signals over membrane. And then we have contractility, the ability to shorten and generate the force. Then because we have contractility, then extensibility, that means ability to stretch without any damage to the arteries. And the final step for the muscle tissue is elasticity. That means have ability to back to the nature, the normal and original shape after being stretched. And when I said keep the sequences one by follow, you cannot start, for example, elasticity and then that you said conductivity, which doesn't make sense as I clear. All right. Now, for the three types of muscle tissue, for skeletal muscle, cardiac muscle, and for smooth muscle, we will cover this in details in a practical lecture with the great senior lecturer Zahra. Now, for skeletal muscle, each muscle is a separate organ or discrete organ and is composed of muscle tissue, blood vessels, nerve fibers, and connective tissue. The three connective tissue sheets or cover, it's around the muscle. I will start from outer and to center. So from outer, it is called epimysium. It is connective tissue. So let me make it clear. It's all of them is connective tissue. So I don't need to repeat that connective tissue. You know that the cover is consists of as a tissue for of connective tissue. Okay, so from outer that surrounded the entire muscle, it called epimysium, while the sheath or the cover surrounded the vesicle, which is a group of muscle, it called perimysium, while the sheath that cover the woolly muscle fibers, it called endomysium. Let me make this as clear. So, and by sample, hold pen in your hand. Then the entire your hand is like epimysium, all right? And the plastic membrane that surrounds the pen is the perimysium. While inside the pen, that's the cover is called endomysium. Um, that we see the next slide. That was said, do you hold the pen in your hand? So all the hand it is the epimysium, and then that the plastic cover for the pen it's like perimysium, and the inside a membrane of the pen is called endomysium. Muscular tissue here is also give you a good example of epimysium, perimysium, and endomysium. Muscle cell or muscle fiber, the same thing. When I said muscle cells is equal to muscle fiber, it's like long, cylindrical cell with multiple nuclei, not just single, and it is situated fibers 10 to 100 micrometer in diameter and up to 30 centimeter long. And when I said muscle cells, and when I said that muscle cells, it's mitotic. What does that mean? It's actually meaning that during when we are growing as a child, the muscle cell undergo to mitosis, which is clear that is, and then the muscle it can grow, and the proportion or the relationship of the proportion to this type of is linked with the tone, with the height, with the weight, and also with the age. So then during adulthood, you will see there's no new muscle cell are generated. That's what does that mean? Muscle cell are mitotic. And as I said, the muscle fiber or muscle cell, we have components of it. If I have normal cell, we know normal cell surrounded by cytoplasm by cell membrane, cell membrane, and here we call it sarcolemma. Okay, it's like a plasma membrane of muscle fiber and transmembrane, potentially charge differences on either side. While for normal cell, inside the cell we have cytoplasm, and now here, because it's muscle cell, we call it is sarcoplasm. It is cytoplasm of muscle cells, and it's stored glycogen, mitochondria, myoglobin, and actin and myosin. For actin and myosin, we will cover that soon. Then we have the transverse tubules. It's two tubules. It's not a tubules. That's continues with the sarcolemma that we connect with the cell membrane, and then it extends into the sarcoplasm. That means it extends to the cytoplasm, and it is filled with the fluid. Myofibers is like parallel, is extended length of muscle fiber. Each muscle fiber contains hundreds to thousands and is composed of myofilaments, either thin is actin or either thick it is myosin. Also, the component of muscle fiber or muscle cell is sarcoplasmic reticulum. What it's called is in a muscle cell is endoplasmic reticulum. It's a smooth endoplasmic reticulum, a tubular network around each myofiber, and the terminal cisterna. Cisterna is like flattened membrane, and vast membranes connect between the endoplasmic reticulum and Golgi apparatus. It's like a storage or tank for calcium. And we have another term for muscle fiber or muscle cells. Is it ray? Triad is like a two-tuple with a two-terminal cisterna. Here I have a cartoon diagram. I give you clear what does that mean. Sarcolemma is the cell membrane, and then that we have sarcoplasmic reticulum. When you go to triad, you will see we have two-terminal cisterna. If I said cisterna, that one is in a singular. While in the plural, we called it as cisternae, and then with the two, what with one two tuple. All right, here I have the levels of structural as organization. We carried this sketch from Tom the bottom and like skeletal muscles, um, just like above physicals. Skeletal muscles are surrounded by epimysium. It's like made above fascicles, and this fascicles is surrounded by perimysium. And then that we have the fascicles. It's made above for muscle cell or myofibers, and because the cells are surrounded by sarcolemma, and it is endomysium. Then the muscle cells is made above myofibrils, which it is inside the cytoplasm, we call sarcoplasm. And then we have sarcomeres, it's also inside the myofilament and sarcoplasm. This is from top to bottom. And you can also do it from bottom to top. It's like you can say that sarcomeres or myofilament together to become myofibrils, and myofibrils group together to form or to become muscle cell group together to form vesicles, and vesicles group together to form a skeletal muscle. Whichever side you can read it, but keep it the same sequences. Right. Now the proteins of muscles. Myofibers have three kinds of protein. It's either contractile proteins like myosin and actin, and or regulatory proteins, which is during the contraction on and off, like troponin and tropomyosin. Or it is like structural protein, which provide the proper elements and elasticity and extensibility, like chitin, um, titin and myosin, nuclein and dystrophin. Here I have cartoon diagram. If we take this part from muscle and magnification, you will see muscle bundle. And then the muscle bundle together form the physical. And that physical is about the group of muscle, and you will see muscle cells or muscle fiber. Then that continue by magnification process, you will see myofibril, and myofibril have sarcomere. Let me give you define about sarcomere. Sarcomere is about complicated repeat unit in saturated tissue, and this is between two Z line. You will see this coming soon. All right, this is the slide what I said. If you see the sarcomere in diagram C, you will see it's only between the two area for Z disc or Z line, and it's have two saturations, um, two saturate area. Um, the light areas is like H zone, which is thick filament only, and an I bands, which is thin filaments only. While the dark A bands is like thick and thin filament overlap, while the M line it's the only in H zone. And uh, here it's very clear that the thin filament is for actin, while the thick filament is for myosin. Nerve and blood supply. Each skeletal muscle is supplied by the nerve, artery, and two veins, and each motor neuron supplies multiple muscle cells, neuromuscular junction. And muscle cell, we will see in nervous tissue. We have one motor neuron, a terminal branch, and its contact one to two capillaries. But in general, nerve fibers and capillaries are found in endomucin, and and it is between individual cells. Here I have the proteins of muscles. It's myosin. I start by thick filament, which is composed of myosin. Myosin as the structure is it looks like the stick for golf game. It's the same stick with the two head twisted together, and this head is connect with the thin filament. I mean thin filament with actin, and it is this is structure between two Z lines. Then we have the thin filament, that the thin filament together form actin, and it is touched by the two heads of myosin connect with actin, and it's made of actin, troponin, and tropomyosin. And this thin filament is is only in the Z line and to the next as the sarcomere. Because as I said, sarcomere is complicated repeat unit in saturated tissue. Now I finished the for muscle tissue. Now I start for nervous tissue. Nervous tissue you can give overview is like the head of master controlling the communication system of the body. And the cell is communicated during the have electrical or chemical signals. And these signals is very rapid, specific, and is do it like immediately as a part of second. Nervous tissue have two functions, and very essential: monitors and regulates the functions of the body. Brain, spinal cord, and nerves are composed of the nervous tissue, and they are specialized for being stimulated to transmit stimulus from one a part of the body to other rapidly. While the functions of nervous system, it's either sensory function, that's to sense changes the environmental, either the internal or external environmental, because there is a sense of receptors. And the sense of it is afferent, neuron self-destruction. Or the other function of nervous system, integrative function. That means analyze to the sensory, make decision, have some social aspect or some behavior. And this is linked to interneurons serve this function. Motor function is thus response to stimuli by initiating the action. Motor efferent that neurons serve this function. Here I have two things: afferent nerves and efferent nerves, like comparison between them. See clear to note. Now, nerve cell types and histology. We have two types of the cells of nerve tissue. It's neurons and neuroglia. Neurons is very limited ability to regenerate, no mitotic ability. If we have injuries or have accident, the nerve tissue will be very severe. The conduct of impulses or the stimuli is from one part to other part during the body. But the process is for to have very active transport process mechanism. It depends on the copious of ATP, adenyl the structural of typical neurons is start by cell body, and we have axon, and the cell body is like have single nucleus with the with the nucleus, and we have muscle body and endoplasmic rough, also for ribosome and the protein synthesis. Neurofilaments give the cell shape and support, and also microtubules move material inside the cell. Then we have axon and the a very long tube and the end by axon terminal. You will see this the next slide about myelin sheath or unmyelinated sheath. The cell processes is dendrites and an axon. Well, the main function for neuroglia, it's a supportive and protective cells can go through mitosis and also surround other nerve cells. We have four types of supporting cells in central nervous system, um, and uh, and also we have two types of supporting cell in peripheral nerve system (PNS). For central nervous system, we have astrocytes. We see here in cartoon diagram, it's most abundant neurons that extensions that wrap around the neuron and involve it with the forming the blood-brain barrier and a selective barrier that regulate the chemical environment of the brain. So it's have function or rule of a regulated brain function. Where microglia, it's when we have macrophages in immunity system. Here we have microglia, and it is the same rule: have it to engulf, destroy the pathogens inside the breast. While ependymal cells is like isolated columnar cells that line the verticals, and its cavities to the brain, which contains the cerebrospinal fluid. And the current created by beating of the cell circulate in cerebrospinal fluid. Oligodendrocyte is like have many extensions, myelinated axons of neurons in central nervous system. While for peripheral nervous system, we have two type of cells. Schwann cells that it's form the neurolema, as we have sarcolemma, I mean the cell membrane. So neurolema is the cell membrane for nervous system or nerve cell, and from neurolema, and it's help the role for a regeneration, um, a regeneration of damage in nerve fiber. This is the main function. While for satellite cells, it surrounds the neuron body, and it is provide electrical insulation and regulate chemical environments of neurons. I have cartoon diagram about the nerve fiber structure. As we see in the muscle fiber, here I have the epineuron, which is surrounded the entire the nerve fiber, and then we have perineurium. It is surrounded the vesicle, which is a group of fiber, and then that we have endoneurium at the end of surrounded by each fiber, and continue to the long axons until the terminal. Nervous system in general divided into principal parts, either CNS, which is the brain and spinal cord of dorsal body cavities, which is integration and control center, while a peripheral nervous system, it is portion of as a part of nervous system on our side, and it consists mainly of nerves that extend from brain and the spinal cord. So if it is extended from spinal nerve to and from spinal cord, or it is cranial nerves to and from the brain. Here I have a slide to give you a comparison between CNS and PNS, and you will see it's very clear for human body, the purple one or the pink one, it is central nervous system only to brain and spinal cord, while the blue one is peripheral. You see many extended from it. Now we have myelinated and unmyelinated axons. Schwann cell myelinate, that is wrap or cover around the axons in the peripheral nervous system during fetal or during fetus development. Schwann cells, as the normal cells, have cytoplasm, nucleus forms, and its outer layers we call neurolemma, with the inner portion during the myelin sheath. The tube is diagramming the axons and are that are repairing themselves, and the it's like long tube to guide the ampullus. Here I have a cartoon diagram. If it is non-myelinated axon, they're not covered by sheath of myelinate, or it is the second one is myelinated. And we know that very well. If it is myelinated, that means the action of transmit the ampullus is 30 times will be faster. If it is non-covered, that means 30 times is slower. Here I have very clear that what does exactly myelinated axons. If it is myelinated axis, that means I have a series of Schwann cells lined up along with the axon, and each having proper coating of myelin insulating the axon. While if it is unmyelinated axon, now we see there is the Schwann cells cytoplasm, but there is no wrap coating of myelin surrounded of axons. That is the exactly define if there is myelinated axons or unmyelinated. And also I give a nice table and very easy to know what is the start by define. If it is myelinated fibers, that means it is covered by sheath of myelin sheath. If it is unmyelinated, so it's obviously clear. Myelinations, that's being present, and this is absent. For the ampulluses or the signals or the stimulus, for if it is covered by myelin sheaths, it's faster than it is non or unmyelinated, will be slower. And the nose of Denver is here present, and now for unmyelinated is absent, while for color is changed white for myelinated and unmyelinated is gray. Before I finish the lecture, I go through to go by each slide, and that slide will give you guide, which exactly that's important to know, and which one is good for knowledge. So, um, um, for fraction of muscle tissue, this slide is nice. I need to know very well. Properties of muscle tissue. Keep the sequences one by one, and if you have the nice defined for each term and the sequence is wrong, that means your answer is also wrong. So the three types of muscle, just keep like knowledge. You will covered by great senior lecturer Zahra, and then that each term you need to know about the skeletal muscle. This is only for the for knowledge. You need to know that it consists of the muscle cell, sarcolemma, sarcoplasm, transverse tubules, myofibrils, sarcoplasmic reticulum, and striate. This is only for knowledge. The sketch here is very important. You need to know it. If you write it from top to bottom or vice versa from bottom to top, okay, that's fine. But keep the same sequence. Don't change it at all. The three types of proteins, okay. Give me one example about each protein. For example, if you said contractile protein, give me myosin or actin. If you say regulated protein, give me one, troponin or tropomyosin, and so on. This is only for knowledge. You need to know the light areas and the dark areas. This is also, you need to know how the muscle cell is surrounded by nerve, artery, and two veins. For myosin and actin, please just keep as explained, and do not to focus about the diagram. Okay, just explain it. And for nervous tissue, you have the first slide just for overview or explain. And then the function of nervous system is important. Afferent and efferent, this is also important. And then that the histology of neurons. The 4 types for CNS and the 2 types for PNS is important, and this is only for knowledge, and this is also important for without human body. And this slide also you need to know as explained. This is very important as it defined what the difference between myelinated and unmyelinated. This is a table is also important. Be safe and goodbye. Thank you.